All right, let's go ahead and get started. We'd like to go ahead and get started now. So, this afternoon's speaker is Doctor. I just came up with that. I don't know. Michelle Kahlenberg. She's got an MD PH.D. How many of you planning to do an MD PH.D? Okay, so you might want to talk to her about what you have to go through about doing an MD PhD. It's a very ambitious program. So the topic for today deals with lupus, and part of the reason why I searched out for you is because I do have a friend that I think might be online. Told her to check it. I think she has the SL type of lupus. I don't know what all the different types are. So that was one of the motivating factors for me to search for this topic. She'll be looking at new therapeutic opportunities for lupus. So Dr. Kahlenberg is an assistant professor at the University of Michigan. She's in the area of internal medicine, and more specifically in the division of rheumatology. She did her bachelor's in biology at Denison University, which is a small school, 25 or so hundred undergrads or something of that type. That's in Ohio, and then went on to do an MD PhD at Case Western University in Cleveland. Following that, did a fellowship at the University of Michigan, and then stayed there, and now is assistant professor there. So this is an area of focus, and she'll talk to us more about this. She's really an up-and-coming star in this field, and has received a number of awards already, especially given that she began here in 2012. So it's a pleasure, an honor to have you here with us today.
All right, you guys are scared of me. You're all like sitting in the back. Thanks for inviting me. It's nice to have a look around campus, and I appreciate that the weather was cooperative for the drive over on 94. I think every single person I met today asked me how was the highway, so. So thank you for the introduction. Like Dr. Murray said, I am an assistant professor in rheumatology. I spend 80% of my time doing translational research in lupus, and I spend 20% of my time seeing patients. So I do have a good mix of clinical exposure and research, and I'd be happy to speak with anyone after who's interested in this type of career path. So because I'm a translational researcher, when I was asked to talk to a chemistry lecture, I was a little confused. But I've tried to pick out some topics in the newer areas of lupus research that actually do incorporate quite a bit of biochemistry into the research. And so hopefully this will bring in some chemistry into lupus. And at the very end, if we have time, I'll tell you a little bit about some of the stuff we're doing in our lab. So what is lupus? So this may be what he was referring to, which is the SLE. So we call it systemic lupus erythematosus, or SLE, or lupus for short. It's really a fascinating disease, and it's thought to be one of the quintessential diseases of autoimmunity. Basically, we think the reason that people get lupus is a mix of genetic and environmental influences, and you have to have both. Very rarely are genes sufficient to do it by themselves. And what happens is that you develop autoreactive B and T cells. So these are your adaptive immune cells, and they start to recognize self instead of foreign. And these then attack your organs, and you also, because you have autoreactive B cells, you make antibodies against self-proteins. And these antibodies can then deposit in the tissue in the form of immune complexes. And so you get immune complexes put down, you recruited inflammatory cells, which damage the tissue, which causes more parts of yourself to be exposed. And then you get more antibodies brought in, and you bring in more inflammatory cells, and you get this vicious cycle of immune complex formation, tissue damage, and inflammation. The main test that most people have heard of is called the ANA, the anti-nuclear antibody. And this is an antibody that these patients make which recognize nuclear proteins. So if you take some liver cells and just put them on a slide and then incubate the patient's serum with the cells, and then you come back with a detecting antibody, you can see staining. And the patterns can be different, but we see all three of these associated with lupus. And I also want to point out, as it's actually a very important area of interest for me, is that it's not only the adaptive immune system, but also the innate immune system, meaning the first response, your neutrophils, your inflammatory proteins that are made by things like the skin or the gut. These types of immediate responses that don't create memory are also very important in activating lupus. So what happens to a patient with lupus? So it's a rare disease, but it isn't uncommon. We see it in about 0.5% of the US population is thought to be the prevalence. And one interesting aspect of lupus is that it affects women to a much greater extent than men. The ratio is about 10 to 1. And it affects women primarily during their reproductive years, so from the ages of about 20 to 40. And it can be very devastating because if they get really sick, it impacts their ability to work, to have children, to take care of their families. So these women and men can develop bad renal disease, and this is the #1 cause of mortality in lupus. So they can get immune complexes. This is a picture of a glomerulus in the kidney, which is the filtering part of the kidney. And this glomerulus is full of immune complexes here, which are lighting up with antibodies that recognize autoantibodies. You can get scarring, skin lesions, and photosensitivity. So they can get bad rashes sort of all over the place. They can get a lot of hair loss associated with these rashes if they're on their scalp. And by photosensitivity, we mean these rashes can be brought on by UV light exposure. So they have to be very careful in the sun. They can have drops in any number of their blood counts. They can develop really deforming arthritis. This is an x-ray of a patient with Jacquard's arthropathy from their lupus. You can get brain involvement. They can have confusion. They can have psychosis. They can have obliteration of their spinal cord and become quadriplegic from involvement of their nervous system. They can have something as simple as sores in their mouth. They can get eye inflammation. And one interesting aspect is that there's a 50-fold increased risk of heart attack and stroke in these patients. So we think of the beginning of lupus and the acceleration as sort of a disease spiral where you have genetic predispositions, and these are acted on by environmental triggers. And what these triggers are, we're still trying to figure that out. We think, obviously UV light is one of them, probably things like various environmental hydrocarbons, mercury, other exposures can also help push this along. And activating the innate immune system is probably one of the first triggers. And then this causes the activation of the adaptive immune system. You get the B cells to make the autoantibodies, and then you continue down this spiral and eventually ending in tissue damage. The other piece of this is that patients aren't always sick. Their disease can flare and remit. Usually in order to make them better, we have to give them medications to suppress their immune system. And they can be stable for quite some time before they have another flare. We look at markers in the blood to determine whether or not a flare is occurring. Obviously, their clinical presentation will be one way, but you can also measure things like whether or not their antibodies are going up or whether complement, which gets deposited in the tissue with immune complexes, whether that's going down. And like as I mentioned, the flares can come in many ways. And just because a patient had one manifestation the first time doesn't mean they're not going to have a different manifestation the next time. So you have to keep a very open mind when a patient tells you they're not feeling well. We're really not good at predicting when flares will occur, and this is something that is interest of mine, is if we could predict when patients are going to flare, maybe we could get away with treating them with much less medication before all of the organ damage starts to take place. And as I mentioned, the triggers for the flares include UV light, viral infections, environmental toxins, and we're still trying to tease this out in terms of understanding exactly what promotes lupus. I just put in some more pictures here so you guys can understand how devastating this can be. One of the faculty I met today, they were like, "Is lupus real?" And I was like, "Yes, it's very real." There's a lot of jokes because House was always, saying it's not lupus, but we do see quite a few patients who are very sick with this disease. And so this is an example of the alopecia that can occur. You can imagine how devastating this is for young women to have a giant bald spot like that. This is a picture of a lung. This is a CT scan of a patient who has inflammation in their lung from their lupus. Lungs on a CT scan are supposed to look black like this, and you can see this is full of inflammatory infiltrates. So this patient's going to have a really hard time breathing. You can get lesions in the blood vessels. So this is an example of an ischemic finger lesion because the blood vessels have spasmed, or you can get blood clots within those vessels, which cause the ends of the fingers to die. You can see the same things on the toes. And I also just wanted to mention that lupus also can be passed to unborn children because the antibodies cross the placenta. If you have a mom with lupus who has certain kind of antibodies, they can cross the placenta, and when the baby's born, they can actually have lupus. So they'll have rashes. Sometimes they can have difficulties in their heart conduction pathways. But the interesting thing is when a baby gets lupus from their mom, because the autoantibodies go away by about 6 months because the baby starts to make their own antibodies, the lupus will go away. So this is a picture of a child who was born with lupus, but by 6 months of age, it had resolved. So how do we treat lupus? We're really not that great at it yet. Most of our medications are really more like chemotherapy, and our patients get a lot of side effects from the medications they have to take. If they've got a really active lupus flare, we give them cytotoxic medications. These typically target the autoreactive T and B cells. It basically kills rapidly dividing cells. So the T's and B cells are stimulated and making more and more of them, and so the cytoxin comes in and damages DNA and causes those cells to die. So cytoxin is our biggest gun. We also use one called mycophenolate, which also inhibits cell replication. Methotrexate is another one that we use especially for arthritis and skin disease, which also blocks cell replication. These all have a lot of side effects. They make patients feel really crappy. They can cause liver damage. They can cause infertility. So, we have to do a lot of counseling, and they're not the easiest medications for patients to want to have to take. We also use anti-malarial medications. This was discovered about 50 years ago that people who were taking malaria prophylaxis, certain types of anti-malarial prophylaxis, had less arthritis symptoms. And so we now use anti-malarial medications, one called Plaquenil in particular, as a basic, everybody gets that drug because it's good at blocking activation of autoreactive T and B cells by inhibiting loading of the MHC peptide in the endolysosome. And also, there's a newer medication called belimumab that's been on the market for a few years now, which is an antibody against B-cell growth factors. It works okay. It's not definitely not the panacea everyone was hoping for, but we do use it in select patients with some benefit. So that's really our armamentarium for turning off this adaptive immune response. As I mentioned, the innate immune response is really what probably triggers all of this to get started. And we target this also with this same anti-malarial medication, Plaquenil, because it also inhibits endosomal toll-like receptor signaling. So these are very important pathogen recognition receptors that are within the cell, and they bind to DNA and RNA. And if we block this signaling, we can also have some benefit on flare rates in lupus. And right now there's studies looking to see whether we could prevent the development of lupus in people who have ANA antibodies but haven't actually developed lupus. So there's some interesting trials going on here. And then I also put sort of in a lighter color here the anti-interferon receptor antibodies. So these are in trials right now. There was actually some really exciting data in November presented at the American College of Rheumatology meeting showing that antibodies that block the type 1 interferon receptor actually have pretty good therapeutic efficacy for rashes. We're waiting on more data for more severe manifestations, but for the rashes, they seem to be good. So I wanted to bring this up because much of the rest of my talk is going to talk about interferons and how we're discovering how mutations can relate to them and how different signaling pathways within the cell are resulting in interferon signaling upregulation. And so this is really an exciting area in terms of where lupus therapies are going. Okay, so there's basically three major new developments I wanted to bring up, and all of these have a little bit of biochemistry in them. So the first, which we'll spend most of our time on, is looking at these nucleic acid signaling pathways and how they activate these interferon responses. We'll also talk a little bit about epigenetic regulation. This has become a very hot topic in lupus research, and how we turn our genes on and off seems to be important in the development of autoimmunity. And then at the very end, I'll talk a little bit about activation of IL-6 pathways in the skin. This is data from my lab that we're just getting ready to publish. So what is nucleic acid signaling? So obviously, RNA and DNA are the two nucleic acids, and we have machinery within our cells that can respond to RNA and DNA when it's not in the place where it's supposed to be. And when we activate these pathways, it results in upregulation of antiviral responses. And these are primarily driven by these type 1 interferon pathways. The other side of the activation is also inflammatory responses. So you can get activation of NF-kappa B pathways. This is a main regulator of pro-inflammatory genes. And then this turns on pro-inflammatory cytokines like IL-1 beta, IL-6, and TNF-alpha. So you get both interferon responses and pro-inflammatory gene responses, but the interferons seem to be really important drivers in lupus. So as I mentioned, these interferons, they're a group of cytokines that interfere with viral replication within host cells, and that's how they got their name. And there's been more than 10 interferons described in humans. And they really are what make you feel sick. So when you get a cold and you have that first day where you just don't want to get out of bed and you're just like, ugh, that's your interferons being turned on as part of your antiviral response. They really make you feel crappy. And so that's one of the reasons why lupus patients really feel bad when they have a flare, because their interferons get turned on and they just don't feel well. I put some pictures up here showing how these signal. So there's many type 1 interferons, and they all act through this type 1 interferon receptor to activate JAK-STAT signaling. STATs are transcription factors that can then activate these interferon response elements and turn on what we call these interferon response genes. Type 2 interferons, interferon gamma, also signals through JAK-STATs but activates a slightly different STAT pattern and can activate other transcription pathways. And then these type 3 interferons are still being characterized. They also activate JAK-STAT signaling, but how they act downstream is still being worked out. And whether or not they contribute to lupus at all, we're not sure. So as I mentioned, type 1 interferons are really the unique ones to lupus. We also do see some type 2, but type 1 are the main focus in lupus research. And they can have a lot of different effects on the immune system. And if you think evolutionarily, these all would have been derived to have antiviral fighting effects, but if you've got autoreactive B cells or T cells, they can promote autoimmunity instead. So it causes B cells to proliferate and to class switch to make IgG, which is the sort of terminally differentiated type of antibody, and most autoantibodies are IgG subclasses. So it can sort of make a B cell that maybe had an IgM autoantibody into a more pathogenic IgG. It can turn on macrophages and dendritic cells, which are important in the innate immune response, to actually present antigen to these autoreactive T and B cells, so it makes them better antigen presenters. And it also can promote expression of inflammatory cytokines, which go back and activate the macrophages and dendritic cells and the B cells and the T cells and sort of keep the whole thing going. The other interesting thing that I did some work on when I had first started back in the lab was that these interferons also have profound effects on vascular health. And we think that one of the reasons for the increased risk of heart attack and stroke in lupus is because of the effects of interferons on the vasculature. So how do we know that interferons are important in lupus? They have evidence, lupus patients have evidence of turned on interferon genes in many places. So if you look in the blood, in the kidney, in the skin, you see these interferon genes all turned on. We use mouse models to help us dissect, things that are truly pathogenic in disease. And if you knock out interferon signaling in mouse models of lupus, you basically abrogate disease. And also, if you give interferons to young lupus-prone mice, you actually make lupus happen faster. And so we think that interferons are important because if we take them away, you're protected. If you give them, you get worse. And we see these interferons in lupus patients. And now with our clinical data, we can show that if you block these pathways, the patients actually get better. I just wanted to put a little brief mention up about the skin. We know that type 1 interferons are very highly upregulated in cutaneous lupus. So here's a picture of a healthy control piece of skin. This is the epidermis, where the keratinocytes, or the basal layer, these are the cells that make the top layer of the skin, and then these differentiate and die and form like this bridge of dead cells, which actually is the outer layer of your skin. And then underneath, normally there's no inflammatory cells here, and you've got nice tight packed collagen and healthy skin. In 2 different kinds of lupus rashes, you can see there's staining here for MX1, which is the interferon response gene. So you can see there's tons of evidence of interferon being turned on in the epidermis and in the dermis, and you also see all these inflammatory cells coming into the dermis, which causes it to look red and to itch and to swell up and be very uncomfortable for the patient. When we look at genetic risk factors for lupus, one interesting thing that we see is that many of the lupus risk genes actually involve activation of these interferon pathways. And I wanted to point out this molecule here, TREX1, which we're going to talk about next. This is a risk factor for lupus in humans. Other genes like STAT4, TLR7, this is an endolysosomal receptor for RNA. This also has risk factors. IRF5 is a downstream transcription factor from type 1 interferon. So all of these genes, if you have mutations, can increase your risk of lupus. So how do we make interferons? And how would these mutations in these pathways actually contribute to lupus? So one of the ways we make interferons is these endosomal toll-like receptor signaling. So I mentioned this before, that TLR7 and TLR9 are 2 receptors that sit in our endosome, and they bind to RNA and DNA respectively. And when they come in contact with nucleic acid, they then signal to turn on both NF-kappa B pathways, which drive inflammatory cytokines like IL-1, IL-6, and TNF, and also to turn on the interferon genes and make interferon alpha and interferon beta, which are the 2 main type 1 interferons. Where does this nucleic acid come from? Well, the whole system was set up to respond to viruses, right? So if a virus is in your cytoplasm, you can shuttle it through the endolysosomal pathway, or if it was taken up by endocytosis, you could get DNA or RNA in the endosome. Same for bacteria. You get their DNA into the endosome this way. So the system was generated so we could respond to foreign. But if you've got a bunch of dying cells, say you got a bad sunburn and all of your skin cells started to die as a result of the sunburn, and you've got all this nucleic acid hanging around, this could also signal through this pathway. And so regulation of nucleic acids and the presence of them is very important, because if you have nucleic acids present, you're going to respond to them. So we have to keep them in check. One of the ways we do this is with a molecule called DNase 1. This lives extracellularly and degrades extracellular DNA. This is actually a risk gene, so if you don't have DNase, if you have mutations in DNase, your risk of lupus goes up. Lupus patients make autoantibodies against DNase 1 and make it non-functional, and so then they have less functioning DNase 1. They're going to have more nucleic acid signaling. We find that if you lose these ability to degrade nucleic acids, your risk of lupus goes up. Another way that we can respond to nucleic acids is with cytosolic DNA sensors. So this is a very hot area of research right now. These have been recently discovered in the last 4 years or so, and a lot of papers are coming out looking at STING signaling right now in autoimmune diseases. So STING is a sensor for cytosolic DNA, and when it gets activated, it also results in activation of type 1 interferon responses and NF-kappa B, so you get upregulation of pro-inflammatory cytokines. And mutations in the STING pathway, including IFI-H1 and in STING itself and in IPS1, all can increase your risk of lupus. TREX1 is the other one that I had mentioned before on that gene list. This is actually a DNase, which lives within the cytoplasm, and its job is to degrade extra DNA that's floating around in the cytoplasm from viruses. But if you lose TREX1 or have a mutation that makes it non-functional, you're not going to degrade the cytosolic DNA very well, and you're going to turn on these STING pathways, upregulate your interferons and your pro-inflammatory cytokines, and this can also promote autoimmunity like lupus. There's some other interesting signaling molecules that also can activate these cytosolic receptors. So bacteria make one called cyclic di-AMP or cyclic di-GMP. These are produced by different bacteria. So if you engulf a bacteria and take it up into your endosome, these mediators could be released into the cytosol and activate STING pathways. So this may be one way that bacterial infections could influence a lupus flare. If these bacteria are causing these mediators to be released, it could activate all of these downstream pathways that we think are important. So biochemistry. There was recently a paper in April which actually published the crystal structure of how TREX1 interacts with DNA. And what they found was that one of the very common mutations that's seen in TREX1, they characterized it and they found that it's only able to chelate one magnesium ion instead of two. And so where it binds to the DNA and actually wants to open it up so that it can untwist it and cut it, it can't do that because it can only chelate one magnesium ion. So some biochemical pathways have helped us understand how these mutations might actually be affecting the function of the enzyme itself. And this paper went on to show that if you make this mutation in TREX1 in mice, this particular D to N mutation, you get production of autoantibodies. You get more immunoglobulin made, this IgG, and double-stranded DNA is one of our specific lupus autoantibodies. And you can see that in the mice that are homozygous for this mutation in TREX1, that their amount of double-stranded DNA antibody goes up. And if we look in the kidney, this isn't a very good picture, but you can see that you also get these immune complexes like I showed you in the human lupus sample. You can get immune complexes in the kidney, suggesting that just this single mutation in TREX1 alone is sufficient to give us lupus-like disease. Other pathways, so there's also another sensor called CGAS, which can activate STING, and they showed that CGAS is also required downstream of mutations of TREX1. So again, here they had TREX1 knockout mice, so they're completely deficient in TREX1, and you can see that these mice get sick over time and die. But if you mutate CGAS, which is downstream of TREX1, you can abrogate this. So you need CGAS present to activate STING. Whoops, sorry. You need CGAS present to activate STING in order to get all of these interferon pathways turned on. And they were able to show the same was true for autoantibody formation, not just survival. They looked also downstream of a DNase 2 mutation. These mice also get a very bad arthritis, similar to lupus patients. And if you knock out CGAS, you can abrogate this phenotype as well. And then I think this paper was actually a really interesting paper that came out in a good journal. It's called the Journal of Clinical Investigation. It came out earlier this year, where they looked at how patients who have defects in a different gene that regulates DNA RNA function, how that can also lead to autoimmunity. So they looked at patients who have something called Aicardi-Goutières syndrome. So these patients basically have a really early onset of lupus-like symptoms. So these get affected before 1 year of age, and they develop a really bad inflammation around their brain. And one of the primary genes that causes this syndrome is mutations in this RNase H2 gene. And there's 3 subunits to the protein, and mutations in any of the subunits cause dysfunction and cause Aicardi-Goutières syndrome. The main role of RNase H2 is to remove Okazaki fragment primers, RNA primers, during lagging strand DNA synthesis. And it also goes and excises single ribonucleotides from double-stranded DNA. So sometimes we accidentally stick an RNA molecule in the DNA, and so it goes and cuts those out and makes it all DNA again. So if you mutate this enzyme, you get Aicardi-Goutières syndrome, and you also get things that look very much like lupus. You get these same sort of finger lesions, and you can get frank lupus itself. So TREX1, which is the other DNase that we were talking about, this one also causes Aicardi-Goutières syndrome, certain mutations in this gene. So it's probably all on a spectrum of disease, where Aicardi-Goutières is very severe. You get very, very early onset, whereas lupus happens a little bit later in life. So I just put this up here to show you what the brains of these kids look like. they get very severe inflammation and swelling, and then they end up getting scars basically in their brain, and they become very severely developmentally delayed. Most of them don't live past their teenage years. And I just wanted to put up here some of the other things that they get. They get these painful, itchy skin lesions that look just like lupus-type lesions. They can get joint pain and arthritis. They get mouth ulcers. So many, many symptoms very similar to lupus. And so this paper actually asked a really interesting question, and that was, do parents of these patients, so the parents would only have one out of the two copies of the gene mutated, do the parents have a higher risk of developing autoimmunity? So they went and looked at parents of patients who got Aicardi-Goutières syndrome, and particularly looking at patients who had this RNase H2 mutation, and they were able to see that about 35% of the parents had pretty high titer positive ANA antibodies. The general population, just to give you an idea, somewhere between around 5% of the general population, they found a little bit higher, up to 10%, has a positive ANA. So definitely greater than what we see in controls. The parents of these patients had mutations, suggesting that you could get autoimmunity just by having a single copy deficient in this RNase protein. They then went and looked at I think 600 lupus patients and sequenced their RNase H2 genes, looking for mutations in these genes that may have been undetected. And they did find quite a few mutations in these 600 patients that they looked at. And they're going to look at some of these patients who lupus patients who have these RNase H2 mutations and compare them to Aicardi-Goutières patients in the rest of this paper. And this is the structure of RNase H2. You can see the 3 different subunits, and they just mapped here that these mutations basically affect all the subunits together. So one of the things they were interested in looking at was whether patients with lupus who have these RNase H2 mutations have RNA incorporated into their DNA. So they do what's called a COMET assay. Do you guys do this at all in chemistry, biochemistry? So basically the concept of the COMET assay is where they, so when you have pieces of ribonucleic acid stuck in your DNA, it makes it unstable in alkaline conditions. So if you put it in a high pH and then put it on a electrophoretic gel, you will pull some of that DNA out because it degrades. And so you're going to get the DNA sort of shifting behind the cell. So that's what's called these COMET moments here. You can see in the Aicardi-Goutières syndrome patient who has a dysfunctional RNA H2 mutation, when you put it in alkaline conditions, you get this comet tail forming, and this suggests that there's RNA within this DNA. The lupus patients, you can see them, doesn't project very well. In the manuscript, you can sort of see it's definitely not as impressive as the Aicardi-Goutières patients themselves. And they also went and looked at fibroblasts from mice that were deficient in RNase H2, and you could also see these they call these olive tail moments. You could also see those being formed there. So it suggests that you actually are getting functional deficits in the RNase H2 when you have these mutations that are found in the lupus patients present. Then they went to look and see if these fibroblasts from these patients actually made more type 1 interferon when they had these mutations present. And you can see here that the Aicardi-Goutières patients, when stimulated, this is a TLR3, this is also a different endolysosomal TLR, but when they use this stimulation, you get a little bit of interferon production. When you add UV light, however, you get a much greater interferon production in the Aicardi-Goutières and in the lupus patients with these RNase H2 mutations than you do in controls, suggesting that you may have a functional consequence of increased interferon production downstream of these mutations. They looked in the skin of these patients and you stained for MX1, which is, again, an interferon response element, or CXCL10, which is a chemokine upregulated by type 1 interferons. And you can see that you get much greater staining of interferon genes within the skin when you've got these mutations present. And you also see more chemokine CXCL10 present as well. So this, I think, is really interesting because why would they make more interferon in response to UV? And what they showed was that these cyclobutane pyrimidine dimers, called CPDs, they're the most common of most common type of UV-induced DNA damage. And they saw that they actually get increased CPD formation after UV light in the patients who have this RNase H2 mutation. And so you can see here, this is just a way that they measured how much of these CPD dimers there are. So in the Aicardi-Goutières patients and in the lupus patients, you got more dimer formation than in controls after exposure to UV light. And so they looked at this from a biochemical standpoint, and they measured the angles that occur when you've got an RNA next to a DNA, and you can see that the angles actually change, and they felt that this made the cyclobutane pyrimidine dimers more likely to form when you actually had the RNA present in the DNA. So they hypothesized this is one of the reasons for the increased interferon response, because you're going to get more DNA damage and signal these DNA damaging pathways downstream. So I thought this was a really interesting example of how biochemical analysis of this particular mutation is giving us good mechanistic explanations as to why interferons can be higher in patients with lupus. A different paper that came out in a journal called Immunity, which is also another very good journal, a few years ago, also looked at this concept of what happens when DNA is actually exposed to UV light. So a different type of product that forms in the DNA after exposure to UV are 8-hydroxyguanosines, and this is what they look like structurally. So both UVC and UVA light can induce these 8-hydroxyguanosine structures. But I think the really interesting thing that this paper showed was that when they looked in the skin of lupus patients, they could see these 8-hydroxyguanosines present. And then they went on to irradiate DNA and inject it into the ear of a mouse. And they saw that this actually caused a lupus-like lesion just from being exposed to this DNA that had these 8-hydroxyguanosine adducts. And you can see here, so if you don't UV irradiate the DNA and inject it, it doesn't have any change to the ear thickness. But if you UV irradiate the DNA first and then inject it into the ear of the mouse, you get much greater swelling in the ear. This is a histology here to show that mouse ear is really, really thin. So you basically just have a layer of epidermis, a little bit of connective tissue, like some cartilage, and then that's it. But you can see here, if you UV irradiate the DNA first and then inject it into the ear, it becomes very immunostimulatory. And they stained it for CD45, which is a marker of inflammatory cells. And you could see that you're getting a big inflammatory response to this modified DNA. So this is another reason, from a biochemical standpoint, how we could change the DNA to become much more stimulatory to the immune system. And probably my guess is that this has a better effect on stimulating the STING pathways. So to sort of summarize this part of the talk, basically we've got this STING pathway which responds to nucleic acids. And in lupus, there's many ways this can go wrong. You can have mutations which activate this pathway. You can have mutations in genes that try to regulate this pathway by degrading DNA. So if you don't have them present, you get more DNA there to activate the pathway. You can have mutations in DNA outside the cells so that you get more DNA exposed to cells. You can also have mutations that allow for RNA to be incorporated into the DNA, which makes it more apt to become damaged and stimulate the pathway. Or you can have exposures like UV light or maybe other toxins that modify the DNA to make it more immunostimulatory. And all of these things, when they activate the STING pathway, can contribute to something like lupus being developed. Okay, questions? Okay, so we're going to switch topics briefly to talk about epigenetics. This is another hot topic in lupus. And this is when you get changes in gene expression that are inherited from cell to cell, but it's not caused by a DNA mutation. You're not changing the sequence of the DNA, but just how the DNA gets expressed. And one of the ways this happens is by methylation. So if you methylate a promoter of a gene, you basically turn it off. And the reason this is that normally when it's demethylated, it's open. The nucleosomes are sort of far apart, so all of your DNA replication machinery can get into in between the nucleosomes. It's transcriptionally accessible. If you methylate it, however, you recruit in factors that can deacetylate the DNA, and this leads to it being closed off, and it's transcriptionally inaccessible. And interestingly, when you replicate the cell, if something is methylated and closed, it stays that way in the next daughter cell. And so this is actually an inheritable phenotype. So if you have a lot of things methylated, they should stay methylated as you replicate unless there's something that comes along to change that. So for example, in T cells, we use epigenetic regulation to differentiate our T cells into either a Th1 subtype or a Th2. These are both T cells that are derived from naive T cells, but they make different factors and they influence different types of immunity based on what they make. And this is done by methylation. So if a naive T cell gets activated and exposed to IL-12, this results in methylation of the IL-4 gene and demethylation of the interferon gamma gene, so this cell will make interferon gamma, and vice versa. If it gets exposed to IL-4 when it's activated, it methylates the interferon gamma gene and turns it off, and demethylates the IL-4 gene, turns it on, so that this cell will secrete IL-4. So in lupus, we know that DNA methylation is reduced in the T cells of lupus patients, and probably in also other cell populations, but it hasn't been as well studied. And so many genes that are normally silenced become active. And so what one of my colleagues at University of Michigan has found, Amr Sawala, is that actually in T cells, the genes that activate type 1 interferon pathways are hypomethylated. So they're primed and ready to be turned on, and they're easily accessible. So it's very easy to make interferons in T cells from lupus patients. And we know that environmental factors play a role because environmental exposures can influence methylation. So nutrients, for example, folates can influence your methylation pathway. So if you have a really good diet, you're more prone to have less hypomethylation. So one of our researchers at the University of Michigan tells me he eats a ton of fruits and vegetables every day to try to keep his folic acid high. Hydralazine, which is a very common blood pressure drug that's given all throughout the United States, this actually promotes hypomethylation of genes and can cause a drug-induced lupus phenotype where patients get bad rashes, arthritis, they can feel really yucky, they get autoantibodies just from being exposed to this drug. Life itself, stress, lack of sleep, all of these things can influence the methylation patterns within your cells. And importantly, when we follow monozygotic twins who have the exact same genes, their epigenetic differences happen during their lifetime. So it's your exposures that control how your genes are methylated or not as you go through life. So that's important when you think about living a healthy lifestyle and what you eat and whether you smoke and do all of those other things. They can all influence how your genes are turned on and off. Also, if we think about it, if you remember, I said lupus is more common in women. The ratio is about 10-1. And we use methylation as a way of inactivating the second X chromosome in women, right? You only express one of your X chromosomes, not both. And so you have to turn it off. And we do that by methylating the second X chromosome. So if you're hypomethylated, you could be expressing things off of the second X chromosome that normally are supposed to be turned off. So you might get too much of something. And this is true for CD40 ligand and perforin, two genes that have been associated with development of lupus. And this may explain why women have a higher risk of lupus. And also there's something called Klinefelter syndrome, which is a man, but he's an XXY. So he gets an extra copy of the X chromosome during fertilization. And they actually are at much higher risk of developing lupus as well. And their only risk factor is this extra X chromosome. So issues with methylation may explain why some of these risk factors are there. So I want to go back now to this gene RNase H2 because we talked about all this hypomethylation, but there was this paper that just came out last month, which actually showed that if you have a mutation in RNase H2, sorry, I typoed that, it actually caused, they were able to detect increased RNA-DNA hybrids within these patients. So they did this by something called DRIP-seq, where they were able to detect the hybridization of DNA and RNA together. And they showed that in these Aicardi-Goutières patients with this mutation, you have more of these hybrids. But the interesting thing that they found was that in the presence of these hybrids, you got a global hypomethylation like we see in lupus. And so it may be that also defects in controlling how DNA and RNA are segregating together may influence the methylation, and that may be one of the other risk factors that these signaling pathways may be giving to develop lupus. So not only is it activation of DNA damage from having too much RNA incorporated into your DNA, but also having DNA damage that activates these STING pathways, but also now you can influence the hypomethylation status of the cells by having these DNA RNA hybrids within the cells themselves. And all of this activates the STING pathway and upregulates the interferon response. Okay, so I'm already over time. So very quickly, I will just tell you that in my lab, our interest is looking at how lupus skin lesions actually are formed and trying to understand the pathways which cause lupus skin rashes themselves and how this influences the development of autoimmunity. So one of the findings we have found is that IL-6 is actually increased in these lupus rashes. And interestingly, when we look at the keratinocytes, which is that top layer of cells within the skin, if you look at keratinocytes isolated from lupus patients, they're very hyper-responsive to different signals, including peptidoglycan, which is a TLR signal, or also UV light. They release more IL-6 in response to UV stimulation. And what we found is that we can mimic this finding by treating healthy control keratinocytes with interferons. And if we either with TLR ligand or UV stimulate in healthy controls that were pre-exposed to interferon alpha, you can see that we replicate what we see in lupus in terms of making them more responsive and more likely to produce IL-6. And if we incubate lupus keratinocytes with antibodies that neutralize type 1 interferons and then try to stimulate them, we completely abrogate the hyperactive response within the lupus cells themselves. So we're working right now on a model where the lupus keratinocytes have an autocrine type 1 interferon loop. So they're producing interferons that prime them to have this hyperstimulatory response. So one of the next steps in our lab right now is actually looking at these STING pathways and whether or not they're turned on in lupus keratinocytes and how things like UV light, which cause a lot of cell debris to be formed, could actually be promoting hyperstimulation of things like IL-6 within lupus skin. So a very brief summary of my research, but we're publishing all the time. You can Google us. I've got a Twitter handle too. I can tell you if you want. So in conclusion, lupus is a devastating disease. It's really got a complicated etiology, but I think right now, looking at DNA modifications, DNA signaling, and the interferon responses are really our most promising treatment targets out there. And continued research into the etiopathogenesis of lupus will hopefully give us a better ability to prevent lupus, to treat it when it happens, and also cure this disease for these patients. So I just want to thank the members of my lab. Jasmine Stannard, that's whose picture was on those slides from my lab. She's done a lot of the keratinocyte work. I also wanted to shout out to Jason Knight. He gave me some slides in the intro to use. These are my funding sources. And then very briefly, since when I was an undergrad, I had no idea what a physician scientist was and didn't know these careers even existed. So I thought it might be useful for you guys to just sort of see the pathway of how you get to this. If you're interested in translational research or you think this is something that interests you, this is sort of how it works. So you finish your undergrad, and you can basically choose 3 pathways. So you could go get a PhD in sort of some pathologic related field. You could do it in chemistry if you wanted, and that usually takes about 6 to 8 years. And then if you were really interested in doing a translational, like disease-focused career, during your postdoc, which would probably be, you probably do 2 of them, somewhere between 4 to 6 years, you should focus on getting some translational research education. University of Michigan has a program that postdocs can apply for. It's a 1-year training on how to do human subjects research and some disease education, so that if you didn't get that during your basic training, you could get it later on. If you wanted to just go to medical school, you can do that. That takes 4 years. Then you do a 3 to 5 year residency. If you wanted to do a fellowship, you could sort of skip this step if you didn't, but that takes about 2 to 4 years. And then you would have to do some pretty serious research training, probably about 4 years before you were ready to sort of move on and get on the tenure track. The way I did it was I did an MD PhD. That took me 8 years. And then I did a 3-year residency and then a 2-year fellowship. and then I did 1 year as a postdoc, and then I got on to the tenure track assistant professor. So it's a bit of a long haul, but I have to say it's a really fun career because I get to see the patients in clinic, I get to take care of them and see how much the disease devastates their lives. But then I have the extreme privilege of being able to study that in the lab and take what I see are big questions remaining in treating their disease and try to figure out ways to help them and make things better. And that's a very satisfying job track. So I'm happy to talk with any of you after if you have any questions. Thanks a lot for your attention.
So any questions? Right there. Oh, okay.
Hi.
Hi.
I was just wondering, you mentioned pregnant women and lupus. Is there a higher rate of death in pregnant women with lupus? Since can you treat them? Because it suppresses their immune system, the medicine that they
Sure. So pregnancy in lupus, the data suggests that it's not so much dangerous for the mom, but for the baby. And pregnancy outcomes in lupus are not that great. The issue is that because the interferons are so damaging to the vasculature in the placenta, you get a lot of issues with fetal growth, and the placentas are really kind of small and the vessels are not very healthy. And so the babies are often very small. And the other issue with that is that you can get preeclampsia because probably of signals coming from the placenta. And so the preeclampsia raises the blood pressure in the mom, so that can put the mom in danger, and it also puts the baby at danger. So right now there's no way to fix that because we don't have, until these interferon antibodies get approved and studied, we don't have a way to turn off these interferon pathways right now. So immunosuppressants don't really block those pathways. And so if their disease was active in other ways, like they were having kidney inflammation, we can turn that off and give them medicines. Usually we just give them steroids if they're pregnant because the steroids don't harm the baby. But if they have issues with the placenta, usually premature birth is a very common outcome, unfortunately, in lupus. So yeah, it's a huge issue. It's a big area of study right now.
Over here.
Hi.
Hi. I was wondering if there's so once you detect lupus and you're helping somebody treat it, is there any type of way that somebody could prevent from getting lupus, like somebody else who hasn't had it before? Are there any ways that they can prevent it throughout their life before they hit that period where they're available to get it?
Yeah, it's a really good question, and it's a really interesting area of research. So my advice is if you think you have a family predisposition or you have a positive ANA but no symptoms, is to get lots of sleep. Studies suggest that if you get less than 7 hours of sleep a night, your risk for developing lupus, if you're one of those ANA positive people, goes up. Stress can make you more likely. Poor diet, smoking, not exercising, all of those things that increase free radicals and other things. We don't have good evidence, but I think it's smart to try to live as healthy of a lifestyle as you can. Like I said, there's trials looking at these anti-malarial medications and whether they can prevent the development of lupus, but the data's not out yet, so we don't know. If I see a patient in clinic who has a positive ANA and maybe has a little bit of symptoms but not enough for a diagnosis, I'll still put them on the anti-malarial because I want to try to turn off whatever's trying to turn on. And sometimes that helps. So yeah, but we're not good at preventing otherwise.
Hi. So you mentioned that women are already more likely to get lupus than men, but why in particular for the age group from 20 to 40 during the reproductive years is it prevalent during that time than later?
It's a really good question. So we do see it in kids, but those kids probably have some of these more aggressive mutations that have increased their risk. Women who get it between the ages of 20 and 40, probably the influence of estrogen on all of this is important. If you take a lupus-prone mouse and you remove their ovaries before they have evidence of autoimmunity, they'll be protected. So definitely estrogen plays a role. We don't understand fully how, but definitely that's one of the reasons in that mix. So.
Hi. Okay, so you mentioned how this like area of research in lupus, like the DNA signaling and trying to target that and optimize our understanding of it. I was just wondering if there were any like other areas of, I guess, the body physiology that has been researched that was helpful to the lupus research.
So the most commonly studied organ is the kidney. Because that's, probably the most, that's the organ that causes the most death and morbidity if it becomes affected. So there's a lot of trials that are ongoing and a lot of research in that area, trying to understand what are all the cell populations actually involved in nephritis. There's a huge push right now from the NIH in combination with industry, trying to basically understand all of the RNA sequencing signatures that exist in lupus nephritis and all the different types of lupus nephritis. So there's a lot of research going on in that area. I didn't focus on it here just because I was trying to come up with biochemical things to talk about. But yeah, so kidney disease is a big area in lupus research. And we do have some, we can treat and put about 40% of patients into remission and they don't have another flare. It's that other 60% we don't understand why they don't respond as well to the medications and why they have to keep going back on more cytotoxic therapies. And so we're working on getting it. Europe actually has a very interesting study where they rebiopsy patients. And so they look at the beginning and then after 6 months of medication, and then they check them again a few years later to see whether there's smoldering inflammation or whether they can find things that are predictive of response to certain medications. So that area of research is ongoing too.
Hi, I was just wondering if there was genetic screening available before birth for lupus?
No, and the reason is that even though I sort of emphasize these risk factors, the penetrance on those genes is very low. So most people who have these mutations won't get lupus. It's just that if you have them, your risk is higher. There are a few mutations where the penetrance is almost 100%. So if you have a deficiency in something called C1q, you're about 100% likely to get lupus. But we don't screen before birth because we don't have any way to treat it or fix it, and we don't know if it's going to make a difference to know that information before birth anyway. And so it's not considered part of routine practice. Now, if we were able to find, some mutation, other diseases where they have the mutation and for sure that they're going to get it, and if we can have a therapy for it, then you've got a good reason to do the pre-birth screen. But, you can look at your family history and make a guess whether or not you have a high chance of lupus or not,
Is there any other disease per se that is really commonly mistaken for lupus because there are very common symptoms or just?
That's a good question. So we get a lot of referrals when patients just have positive ANAs. And that doesn't mean you have lupus. It means you have one of the pieces of lupus, but not all. Mm-hmm. So we see a lot of patients in clinic and reassure them, right now you don't have lupus. Some patients who have, issues with fatigue sometimes can wonder if they have lupus because fatigue is a very common symptom in lupus. But I try to be very clear about what is lupus and what is not. And I really am strict that if I'm going to call you lupus, you better have something that's going to be treatable with medications that target lupus issues. So if it's just that you're tired and you have a positive ANA, I don't count that as lupus. So, yep. Hi. Uh-huh. Yes. Dude.
Okay. Is it on now?
Okay.
Okay, it's on now.
Okay.
Okay. And so I was wondering, I guess kind of in relation to what Gabe asked about the pre-genetic screening, how do you know to turn off the second X chromosome? I'm guessing, like if you already have the disease, is it possible to turn it off and then reverse the disease, or is that for like somebody's child, like unborn child?
So every woman in this room has one of their X chromosomes turned off, and it's not like the whole X chromosome is turned off. Like you only express one gene off of a chromosome. So you either pick this one to express or this one, right? So you could turn off like the arm of this X chromosome and the other arm of this X chromosome. So it's not like the whole thing is just packed up and put away. And it's all done by methylation. So it just depends on which promoters get methylated on which one. And your cells normally regulate that just fine. But in lupus, because we have things that promote demethylation of genes, you've got this one turned on appropriately, but because we're demethylating all through the cell, you could accidentally demethylate a promoter on the other X chromosome that's supposed to be turned off, and suddenly that's going to express extra gene. And if it just happens to be a gene that can promote autoimmunity, now you've got more than what you wanted to start with. So does that clarify it more?
If a mother contracts lupus between the ages that women are most likely to get lupus, what's the chance that her daughter will also have lupus?
It's a good question. So I think the risk of lupus in terms of genetics is thought to be somewhere around 30%. So how does that translate into numbers? I'd have to look up some papers to tell you. I know the data's out there, but I don't know if I could tell you off the top of my head. So it's probably like, it's about a 30% penetrance, I think, if you have a family member who had lupus. So then that would be like a 1 in 3 chance of having lupus if your mom had lupus. But don't quote me on that. I have to double check my numbers.
Oh, Emily?
So you said that some babies are born with lupus and then it goes away.
Yeah.
So do they have a greater chance or a lesser chance of getting it later in life?
I think you have the sort of like the genetic risk that way, but their lupus was caused just by those autoantibodies crossing over. So that should just go away when those autoantibodies are done. Now, they probably have a genetic risk of lupus because their mom had lupus, but that would be separate from the neonatal lupus. The biggest issue we see is that if they have heart block, where their conduction system in their heart doesn't work as well because of their neonatal lupus, that's a scar. That doesn't get better. So even though they don't have the inflammation anymore, they usually get a pacemaker. So.
Okay, I have one question. I think we could make this a last. Is there any studies relating smoking to lupus or the effects?
So yes, the studies that are on the top of my head are that if you smoke, you have a much higher risk of skin lupus, and it makes the skin lupus much harder to treat. So smoking definitely influences the skin. In rheumatoid arthritis, there's really good correlative data showing that smokers have an increased risk of the more aggressive form of rheumatoid arthritis, and they also have to go on more medications in order to treat the rheumatoid when they smoke. The other bad thing about smoking in lupus is because you have this higher risk of heart attack and stroke because of the lupus itself, and you smoke on top of it, it's like 2 + 2 = 5. And so that's really a bad combination. So we really try to encourage our patients to not smoke because there's so much effect on the blood vessels in lupus, and the smoking just amplifies that. So.
Okay. So very interesting seminar. We want to thank you very much again.
Thank you. Thank you for having me.
Do you have opportunities during the summer for students to do research with you?
In the division, we do take some summer rotons. It just depends on how many I've got at the time, whether there's openings or not. But yeah, you can always email me and see if there's something there.
Okay, good.